slides. You will have to look with this, I'm improvising. So hacking the plants, I call this talk engineering biology. I don't know whether any of you follow news in biotech. I do it for a living, can't really help it. There's this, let's go with new. There's this new technology out there, it's called CRISPR. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. This is a fancy way of saying bacterial immune systems. There's a figure behind me that basically illustrates how this works. So when a bacterial cell encounters foreign DNA, bacteria don't really worry about foreign DNA from anything except viruses, so usually viral DNA. It has a variety of defense mechanisms that involve cutting up this DNA, actually deliberately inserting small chunks of it into the bacterium's own genome so that it can recognize it later. It's acquired immunity. And combined with a variety of enzymes, mostly an enzyme called Cas9, which is CRISPR-associated enzyme number 9. They looked at lots of these, obviously. These small segments of viral DNA get transcribed into RNA, directs this enzyme to a specific point in the DNA that matches the RNA, so that would be the viral genome, and cuts it up. Very clever. Very clever for the bacteria at least, but also very clever for us because we can harness this technology by introducing that same enzyme and a designed piece of RNA into any given cell. We've tested this in hundreds of different organisms at this point, and it works in all of them. So what this allows for is targeted genome editing. It is literally the holy grail of biotechnology because it allows you to knock out a given gene. This is really easy to do, which is great if you're studying gene functionality and relationships between genotypes and phenotypes, which means what does a gene actually do when it's expressed in a cell. It also allows you to knock in genes, which means that if you have a bad copy of a gene that doesn't express, for instance, the enzyme it's meant to express, but a defective one instead, let's say sickle cell anemia is a great example. So rather than expressing functional hemoglobin, it expresses this defective hemoglobin. This is a single base mutation. So you take a single base out of like 3,000, you change it, enzyme is broken. You change it back, enzyme now works again. So think gene therapy here, big time gene therapy. At least that's where all the money is at. The good thing is we can use it for a lot of other stuff as well. So not only hacking bacteria and yeast and so on and so forth, the hack the plants is also a major potential application here. Not least because it means we can engineer things like high salt tolerance, nutrient contents, disease resistance, etc., into crop plants, which means we can grow more crops in less space with fewer pesticides, with less fertilizer. We can grow it in depleted soils. We can grow rice in high salt deltas in Bangladesh. There is almost literally no end to what this tool can do if it's applied right. So to round that off, of course, what are we going to do with it? We'll make glow in the dark mice. What else would you want to do? Make it glow, right? The power of the gods at your fingertips, and you're going to make glow in the dark mice. What else would you do? So instead of you guys asking me questions, my question to you is, if you could change any organism, any disease, anything you wanted in biology, what would you make? What would you hack? What would you engineer? Flying humans! That's more than a single gene, but we can start working on it.
Beastmaster.
It's good, I should start taking notes. Does anybody have a piece of paper? No depends on it.
Your 5 minutes may borrow minutes.
All right, I think my 5 minutes are long up. You have a question?
I have a question.
Yes. All right.
I read somewhere that somebody figured out a way to do this kind of stuff without using bacteria. And
Yes.
I think that helps with like import and export. Can you like explain?
Yes. It is really it is as simple as introducing. So you can cultivate enzymes in bacteria, right? You can engineer bacterium to express basically any given enzyme. In this case, we'll make them express lots of this Cas9 enzyme, which is the one that takes the guide RNA, finds the right place on the DNA and cuts it. Doesn't actually need to cut it, but that's kind of an aside. So you express this enzyme in bacteria, you purify it out, you combine it with a short piece of synthetic RNA, because you can produce RNA through chemical synthesis. You don't actually need a living system to produce it. And you combine the enzyme and the RNA, you introduce it into a cell, a human cell, a mouse cell, a plant cell, whatever, and it works. Which is really the beauty of it, is that the functionality of this technology is not cell or organism dependent. Sorry?
Should we do it in our kitchens?
Should we do it on what?
In our kitchens?
Yeah, why not? I mean, it really is more or less easy enough that you could. You would need to do, you'd need to know basic sterile technique for most cell culturing. Some things are trickier to work with than others, but if you just wanted to engineer ordinary brewers yeast, yeah, you can totally do it in your kitchen.
You said you got like the power of God at your fingertips. So I'm curious, how many people, like other people working with CRISPR, who has the best like mad scientist left? Yeah.
That's a really good question. I think George Church probably. He's a pretty good mad scientist. He's also a pretty good mad scientist. Fucking batshit crazy dude.
Have you seen Spy Kids 2, Island of Lost Dreams? You should watch it. It's very present.